So our speaker today is Dr. Yan from the School of Computer Science at University of Guelph. And she actually mainly works on bioinformatics and machine learning. She has been involved in many prestigious grants. She received a NSERC Discovery Grant, and she has been participated in Microsoft Azure Research Grant, NSERC Create, and Canada First Research Excellence Funds. And today's talk is in the intersection of machine learning and bioinformatics. Dr. Yan, the floor is yours. Please go ahead.
Yeah, thank you. Can everyone see my shared screen, like the full screen of the first page of the slide?
Yes.
Okay,
Yes.
awesome. Yeah, thanks for inviting me. And so let's just jump into the topic as we're a little bit late. So as Curtis mentioned, my research area mainly is on bioinformatics, computational biology. I use machine learning to deal with biological data analysis. Currently, I'm in Guelph, and before coming here, I worked at University of Thompson Rivers University in BC. And I graduated from University of Saskatchewan, and also I did a postdoc in University of Western Ontario. So kind of been to several different places. So for today's talk, I'm going to briefly talk about the research background, and then we're going to talk about two different kinds of biological data. One is called a single-cell RNA-seq. The other is whole genome single SNP data. And we're mainly going to use penalized regression and some combinations of them to do some analysis. And those two research work has been mainly completed by two of my master's students. And so for the beginning, I think we need to talk a little bit about biology and why do we care. So DNA carries the architecture of like those information, and through this transcription process, it becomes mRNA, and then through the translation, it becomes protein. And all those functions, different biological process need to be carried out by proteins and their interactions. So to better understand the mechanisms and how they work and how the genetic information has been passed by DNA and different species or generations, we would like to look at different kinds of biological data. And there are tons of different computational algorithms that have been focusing on different data. And for some of the work I have been doing and my students have been working with, so we look at genome data. So from the DNA sequence, we get some information. And also we look at RNA data. So the RNA-seq is one kind of the data we're looking at. So the first, we're going to talk about the RNA sequencing. So usually, we get a sample from a specific like a tissue or maybe some other parts of the organism that we're interested in. And through some experiment, we'll be able to get their RNA sequencing, and then we know like how the gene has been expressed. And by using some machine learning algorithms, we'll be able to find out how the genes are differentially expressed. And if we look at example of disease treatment, and when we identify some important genes that are significantly different than the rest, then potentially we'll be able to develop some drugs that's targeting to those genes that we have identified. So that's in general the RNA-seq. And currently, the single-cell RNA-seq is a trendy technology. So compared with traditional, that's so-called the bonk RNA-seq, it will be able to look at the RNA expression levels in the cell level instead of like just a tissue level. So that's kind of the main difference. They use like different labeling technologies to do that. And I'm not going to talk too much details on the experiment, but as a computational analysis part, we are interested in what kind of data we will be getting from this whole experiment. So you can view that the data we'll be getting is like a gene expression matrix. And for each rows will be like one cell per row. So we may have different cells, and each column is a particular gene. And we'll get some numbers, and those data are like kind of pre-processed through this whole experiment. But the numbers we're getting here represent their expression level. So the higher numbers, or like more abundance of the genes that we can observe, they're more expressed. So that's kind of this single cell RNA-seq data. And why do we want to analyze this data? So for single cell, it will be able to let us looking at things on the cell level. So a typical application on that is related to cancer study. We could look at different tumor cells to differentiate normal and in different stage of the disease, and comparing their expression levels. And then we'll better understand how the disease has been developed. And by looking at the data, there are some challenges. Usually, we're dealing with high-dimensional data. So if you look at the matrix, we'll be looking at a kind of giant matrix. And the other thing is the number of features, which are genes in this case, usually are much larger than the number of cells. So that will also make it challenging to identify those small set of genes from this big pool, which are differently expressed and also potentially related to like the tissue development. So then we're thinking about what kind of method to use will be suitable for analyzing this kind of data. So one of the methodology that we could be using is regression, penalized regression. Under the penalized regression, there are quite a lot of different methods, like ridge. Lasso is another popular one, and there are a lot of different variants of lasso. And the differences of those regression models are they have different forms of penalty applied. What does that mean? So if we look at a general regression equation, so y is our response variable. If we're thinking like cells is like disease or normal, so that's kind of our y. And x is our expression data, like the single cell RNA data we get. And beta is the coefficient. So that's a general form, and different penalty, a form of penalty will be applied on beta. So that kind of differentiates that penalized the regression. Why penalized regression will be a good method? And because first, it aligns well with the biological reality. Within the huge amount of genes that within that matrix, there are only a small subset of those will be related or causing the states or the disease. Oh. So by applying different penalty, we're trying to limit the number of different genes that having like coefficients values or contributions to the X. So that's kind of from the theoretical part, why that making sense. And the other thing is there are some success on using the lasso method on our single cell RNA-seq data, but not some other penalized regressions that we are talking about here. It is also relatively efficient. Some other method for dimension reduction could be computationally expensive. And when we look at current literature, we don't find a lot of systematic comparisons of different penalized models on the single cell RNA-seq. So with that, we're trying to do a comparison of different penalized regressions. So we have selected 7 different regression models. And the last 2, group lasso and sparse group lasso, they will need additional clustering before handling that into the model. Okay, so that's how we kind of came up to this research topic. And by completing this study, we're hoping to find out how the different regression model perform in terms of their prediction AUC and their computational time. And if we got some knowledge by benchmarking those methods, we're also thinking if the performance can be further improved if we somehow combine them. So this work is mainly led by one of my master students, Babitri, from Thompson Rivers University. Okay, so to systematically compare those 7 different penalized models, we collect some single-cell RNA-seq data. So far we have 4 data sets, mouse, human, and this Arabidopsis theliana is plant data. So when we look at the level of genes, so they're like tens of thousands or hundreds of thousands. But if we look at the number of cells, which are kind of the samples we're looking at, they're relatively small comparing to the total number of genes. And sometimes if we look at the data, they're not balanced. Some are well balanced, but some are not. So there's some additional pre-processing we want to apply before dealing with on that penalized regression models. But that's the data set we have. So when we try that using different penalized regression that has been implemented in R, we look at their average cross-validation AUC to see if they kind of perform well. So something we observe here is result could vary depending on the data you're working with. For some data sets, most of the methods work almost perfect, but some of the data sets, we do have some variations. And another thing we have looked at is their computational time. So from here, in general, we see that reach takes a lot of time compared with some other methods. And we also do see the group lasso, the computational time vary significantly, but if we look at sparse group lasso, it seems relatively stable. We took a further look at of this case, why it took so long. So it turned out that depending on how they group the different genes, when they group genes, some groups of the genes will be assigned weight of or coefficient of 0, and if we have a lot of group genes which being assigned in that case, then that's taking less time for the rest of the computation. So it kind of depending on how the group has been assigned. Some other models also do vary a little somehow. So that sometimes depending on the complexity of the data that we get. So that's kind of what we gain from looking at 7 different penalized regressions. And with that, we're thinking how could we combine them so that maybe we can further improve the performance. So with some design, here is the flow that we end up with. So we selected 4 penalized regression model here on the top, and then we select some top genes from their result, and then do a union. And in that case, we'll be able to narrow down the candidates of genes by kind of this step. And after that, we send the selected genes to the sparse group lasso, which seems to perform pretty well in the previous analysis. And using that, we'll get the final list of genes. So speaking of selecting top genes to form that union, we really also care about the genes expressed differently, and also the number of genes and what kind of genes are those, because that will give us more of an insight of the data. And when we select those genes, so it's just the sort, their coefficient related to each of them. And then the cutoff we set for this study is kind of the mean. So anything above that absolute value of the mean will be selected. So that's how we form the gene pool. So if we look at one particular data set, so we kind of want to understand the 4 penalized regression models, reach, ElasticNet, drop, lasso, and lasso, how their reported genes overlap with each other, and if the union is making sense. So we do see there are some overlap depending on like models. So our purpose is to see if we keep the union, then we will still keep most the potentially contributing genes within this union. And also we don't need to deal with the whole data set of genes, which has totally like maybe tens or hundreds of thousands of genes. So that's for this plant data set. And if we look at the mouse data set, one interesting thing here is we see there's no overlap of drop, lasso with those 3. So keeping all 4 in that forming of gene pools, hopefully will, just keep those genes. We don't want to lose them. Okay, so we then compare our performance of this kind of flow chart, flow with the SGL, sparse group lasso, which we view as one of the best performing penalized regression when we do the benchmarking. So here are our total set of genes. And when we do that union thing, we can see we can significantly reduce the number of genes that will need to be further dealt with. And then this is SGL working on that, getting that AUC and our new algorithm. So they're kind of similar, comparable, and in some cases, a slight improvement. So we think that's this combined workflow work a little bit better. And when we look at our findings, another really interesting thing we find out is that for those reported important genes, we find a subset of genes, which is the pi RNA, as one of the top genes. So this is non-coding RNA, which has unknown function, and it stands out as the top gene that could suggest some important roles, contributing to differentiate the cells from normal to the disease state. So that could be our potential candidate, and for the biologist to further validate. And so the last thing we have done for this analysis is trying to do the clustering or classification of the 2 different types of cells, just using 2 top genes. So there are 2 like pi RNA, like Aiswan mentioned, and on that data set, they can be kind of easily distinguished. But among the 4 data sets that we worked on, not all the data sets show this case. Some data sets can be easily distinguished by just 1 or 2 genes, but others will need more of that. So to quickly summarize on this part of the study, we benchmarked on 7 different penalized regression models on 4 different RNA-seq data sets. And with that understanding, we proposed a new algorithm combining different penalized regression, and it shows a slight better AUC comparing to the top performing single algorithm. And this proposed algorithm also does not require deep knowledge in the grouping of genes. for those group lasso and sparse group lasso. And we also could identify some relevant genes that could distinguish the two types of cells and some potential candidates for further validation. So that's about this one. And so with some success on that, what we're thinking is, can we extend the method to a different kind of biological data, which is also have similar properties of that? So then we come to this whole genome single nucleotide polymorphism data. So this is kind of a genome data. It's not expression. It's more about like DNA or part of that. So what is a SNP? If we look at this figure, you can view this as kind of the DNA of different individual. And on this long chain of DNA, most of the positions, they all have the same nucleotide, but on some positions, they may have different. So this difference of just one single base pair of nucleotide is called a SNP. And if we scan the whole genome, we will be able to identify different spots where different individuals has varied nucleotide. And then that could form as a SNP data set. So each row is one individual and each column is a SNP. And with that, we can know the variations on the genome, but we also need another type of data, which is so-called the phenotype. So a simple example can be disease. So if some individual has certain disease, while others does not. If we're working on plants, there could be some different kind of trees. And with all those two different kinds of data sets, what we could do is we could look at what is the genetic mechanism of a certain trees or certain disease, what causes a certain disease. And how do we know that? We could do an association study between the genotype and the phenotype. And if we use this whole genome SNP data, this is the genome-wide association study, so-called. So another figure kind of briefly illustrate what GWAS is doing. So we have 3 different spots of SNPs, and we have one population in this yellow-brown color, one population in the green. So, in the blue. So, so this group of people are the individuals with a certain disease, and this group of people are individuals without disease. So if we scan on their SNP1, we didn't find much difference. SNP2, we didn't find much difference. But if we look at SNP2, you can see the group of individuals with disease as much more of this kind of SNP compared to the individual groups without certain disease. So those differences could suggest that this change of SNP may contribute or associate with a certain disease. So this is just a kind of a really simple illustration on that. And what we're going to do on this genome-wide study is we're scanning all of the SNPs we get, and then we'll figure out if we can find some SNPs which potentially associated with the certain disease. Okay, so that's kind of what we could use this kind of data to do. If we look at this kind of data, it also has this large number of features. The feature here is the SNPs. If we scan along the whole genome, we'll get quite a lot number of SNPs. But if we look at samples, usually it's also very limited. So large P, small n data. And another thing similar to the previous problem is that the true SNP that associated with a certain disease is also just a really small subset. If we have tens of thousands of SNPs that are being scanned, maybe we can get dozens of SNPs which are associated. So with this data, GWAS has some limitations. Sometimes it misses those true SNPs. They report some false positives and some other limitations. So then what we're thinking is if we could apply the similar technology that we talked before to this kind of problem. And this work was mainly led by my other grad student, Nikita. So we look at how penalized regression performs in terms of identifying those important SNPs. And also if we could combine them to do a combined classifier. So for this study, we narrowed down to 5 penalized regression. The reason for that is some of methods models cannot work for this kind of data. So this is how we ended up with the selection pool. For the experimental data, we have chosen this model plant. The reasoning for that is it is fully sequenced. It has good quality data that are publicly available. Okay, so the data set we have has around 1,300 samples and more than 214,000 of SNPs. So looking at the scale of that is also, we're also having really limited number of samples. In her study, she looked at some other data sets, but right now we're just going to focus on this one and show you some of the results we get. For the type of phenotypes, we have binary. So either this particular plant has anthocyanin or not. And then the germination days is how many days until germination. So binary and quantitative. And if we narrow down to this kind of phenotype, we only can get around 177 samples. And based on that, the different types of phenotype, the evaluation metrics are defined as here. So we first look at this quantitative phenotype germination days. We just look at accuracy, how 5 the penalized regression model work. So when we separate the data to train and test, we look at their accuracy. So we can see here that for the first 3, it's kind of okay, acceptable, but there are some drops with testing that could show potential overfitting. But group lasso and lasso does not perform quite well. And also we can see some obvious overfitting, potential overfitting here with a decrease of the testing accuracy. If we look at the binary phenotype, because we have 4 different metrics, so there are a lot of numbers here. But in general, if we look at, say, ElasticNet, we can see the training test is kind of, in general, consistent. Lasso is kind of similar. Reach sometimes show decrease. And group lasso and lasso will be kind of have larger gap between the train and test. And another thing we have gained from this analysis is that lasso and group lasso will need more bigger machine to run, like we need to have a machine with larger RAM to feeding all the data. Okay, so with that observation, we have tried to develop a workflow that combine different penalized models. So we first feed the data into the reach, lasso, and ElasticNet, and get some important SNPs from those 3 models. Similar as before, we do a union of SNPs, and for the unions, and we will put them into group lasso and SGL to form the final output. So we hope that this workflow could potentially correct overfitting of the performance by group lasso and sparse group lasso. And also we don't need to run it on a big machine, because with the unions of SNPs, we don't need to deal with the whole data set. So we look at this workflow called PandaPen to compare its performance with the group lasso and SGL. So when we look at accuracy, I think that's kind of still acceptable, and we do not see much of the overfitting here. And this table show the similar thing. Different metrics, number looks acceptable, not too much like lower, and also like it's all right for the overfitting, correct some of that. And so I think some people may wonder, when we look at the first three model before, they have also pretty good metrics when we look at their accuracy or some others. But why do we still want to develop the PandaPen? So the reason for that is people care about not only the metrics, but also the output SNPs, because those SNPs will help us to identify the regions along the genome, and if there are some genes that may be associated potentially with the disease. And if we look at the number of output SNPs from the three models here, they're quite a large number, and it's kind of impossible to examine or all of those SNPs reported by them, and to find out which ones are truly associated with the disease or phenotype. And we also mentioned before that in the reality, usually we expect like dozens of SNPs. So that may not be a good pool as well. But if we look at PandaPen's final result, that kind of SNPs fall into what we expect. So that's the biologist will be able to do some further wet lab thing to further validate if those SNPs could point to some genes that are related to the phenotype. On top of that, we also compared our PandaPen with the traditional GWAS. So the GWAS use association study, some association analysis to look at the same thing. And we compare their SNPs to see if we could find those genes and how they're related to the particular phenotype. So we do find a shared gene with our workflow to GWAS software, but for the germination days, we don't find that shared genes. We also find some potential new genes that reported by the PandaPen for the germination days. So this is a transcription factor, could potentially mitigate the process of the seed germination. So that could also serve as additional candidate that worth further study. So when we think about the reasons why those two methods do not agree with each other much, potentially reason could be due with their differences on the models behind. So for genome-wide association study, they usually applied linear mixed model. But this PandaPen, we utilize the penalized regression. And there are also some pre-processing because of differences of models that need to be done. So that may contribute to the less agreement as well. But we find PandaPen could potentially use as a complementary. So maybe combine the two will give us a better set of SNPs that could lead to find out those genes. So finally, to summarize some advantages, so the PandaPen, we can see there is less overfitting in this combined classifier. And another really important thing is the output SNPs is kind of with a reasonable number of the potential list, and we will be able to use them to further validate the results. And this pen leverage the properties of 5 penalized regression models, and so it has some advantage on that side. And to further study those two different topics, we could investigate multinomial classification on a single cell RNA-seq. So for the current experiment, we only deal with the binary case, like there are 2 classes. But in reality, we often dealt with multiple classes. And for the SNPs, as we mentioned briefly, we're hoping to combine pen with GWAS to see if we could have a better reported SNPs or some other machine learning models to find out those associated SNPs. Nikita, my student, she in her thesis, she also investigated Bayesian deep learning model for finding important SNPs. So that's some other candidate method that we could use as well. I would like to thank my collaborators, Dr. Jabat Tamo. So he is a professor in the math and statistical department. So he provides a lot of expertise on the statistical analysis part and the model selections of different penalized regressions. And my 2 students who is mainly conducted those analysis, Bhavitri and Nikita. And our research has been sponsored by Thompson Rivers University and our Answer grant. And also we have used the Compute Canada server to run many of those experiments. So that's all that I would like to talk about. Thank you very much for everyone attending this seminar. And yeah, if you have any questions, I would like to, I'd love to answer them. Thank you.